everyone. So we are meeting again. I'm Sonali Swain and we are going to continue with the next topic of this chapter, molecular basis of inheritance. In our previous topic, we discussed about the DNA. Today we are going to continue with the second topic, the search of genetic material. So it started with an experiment that was given by Frederick Griffith, and he named this experiment as transforming principle experiment. So in this experiment he used mice and also a bacteria that is responsible for causing pneumonia, that is Streptococcus pneumoniae. So he had two variants of this Streptococcus pneumoniae. One that is smooth, which is responsible for causing disease, and the other one that is rough strain. It does not cause any infection even if it enters in the mouse body. So this smooth strain, it has got a coating on its surface which was made up of polysaccharide, and the rough strain lacked this mucus coating actually, and so it used to get identified by the host immune system. Moving further, so what exactly he did was, he tried to infect the mouse with the live S strain, and as he was aware, mouse got infected, because of which it even died. In the other experiment, he infected the other mice with the live R strain, and there also, as expected, the mice remained alive without any infection. Okay. Later what he thought was to kill the S strain. As we are aware, by giving a heat shock, bacteria usually die, and same thing he even performed. He tried to take the inoculum of this S strain and killed it, and later he infected the same heat killed S strain to the mice body, and the result was as expected. Because the S strain was killed, it was no more able to cause any disease to the mice, and so the mice remained alive. And finally, he thought of a different concept where he tried to take the heat killed S strain with the live R strain. So here he was a bit confused. As such, he thought of that the mice will remain alive, as both of these are not able to cause any disorder. But what he found, the result was completely different. He later found that the mouse that was infected with the heat kill strain and also with the live R strain, it got infected and even the mice died. So from this experiment, he concluded that there is something which is getting transferred from the body of this heat killed S strain to the live R strain, and that is why the R strain is becoming virulent and it is able to cause the disorder to the mice. Now it is only possible when there is something that is remaining alive even after this heat kill strain shock was given to. So what exactly happened in this experiment was, the DNA that remained alive even after giving the heat treatment to the inoculum, this DNA got transferred to the R strain body, and that is how it made this R strain to become virulent enough, and that they were able to cause the disorder to the mouse then. So this is known as the transfer of genetic material, and this experiment was performed for the first time by Griffith, and that is where he gave this experiment the name, that is transforming principle. Moving to the next experiment, which was given by the three scientists, and that also characterizes the transforming principle, that whatever the conclusion was made by Griffith, they even proceeded with the same thing, and they wanted to know that what exactly is this transforming material. As Griffith was not aware that it is actually DNA, so the exact identification was made by these three scientists. So here, as you can see, Oswald Avery, Colin MacLeod and MacLean McCarty. So they three worked together to determine that what exactly is the biochemical nature of this transforming principle. Is it a protein or it's a DNA or RNA? because these three things only reside in the cell body and they are responsible to get transmitted and is also available in each and every living cell. So, what they tried to do is, they tried to purify the inoculum by extracting, or you can say by eliminating the other material. So, out of these three, only one was allowed to get transferred to the mice body, and whichever was able to cause the disorder was finally concluded to be responsible to be the transforming one. And that is the genetic material which was getting transmitted from the R body to the S body. So, by this experiment, it thus can be concluded that the genetic material that was getting transferred was DNA, and it was making this R strain to become virulent. So, moving further with this experiment, so here in this experiment, what these three scientists they tried to do, in one of the inoculum, they tried to digest one of the biochemical. And if the disorder was persisting, that means that particular biochemical is not responsible for causing this disease. So, in one of the inoculum, he tried to digest protein by using the enzyme protease. In another inoculum, he tried to digest RNA by using this enzyme RNase. And in the third one, they tried to digest DNA by using DNase enzyme. So, later, what they found was that when DNA was digested by using this enzyme DNase, in that condition, the transformation did not occur. So, this R cell was not able to cause disorder to the mice. And that is how they concluded that it is DNA which is actually working or behaving as the genetic material and is the responsible for concluding this transforming principle. So, moving to our third experiment. So, the third experiment was performed by two scientists, that is Hershey and Chase. It's a most popular experiment where they tried to grow some bacteriophage virus. Now, as you must be knowing, so bacteriophage is responsible for causing disorder to bacteria. So, in this virus, what they tried to do is, they tried to grow this virus in a medium that contained radioactive phosphorus in one of the medium, and in the another medium, they tried to feed them with the radioactive sulfur. Now, as you are aware that phosphorus is a part of DNA, it forms the backbone of the DNA. While in case of sulfur, it is responsible to be the part of protein, as it participates in the formation of disulfide bond in the amino acid. So, this is how he wanted to conclude that it is actually the DNA or RNA that behaves like the genetic material. Now, for that purpose, they started with this experiment. So, what they tried to do is, in one of the condition, they prepared this solution, they prepared the inoculum and they tried to infect the E. coli bacteria. So, after infection, after few time, after letting them to cause the infection, this E. coli cells were like gently agitated by using the blender, which helped in the removal of the virus particle from the bacteria. So, this culture was then centrifuged. Now, as you are aware, in centrifugation, things get separated on the basis of size. So, virus being the light-weighted one was found in the upper region, and bacteria being the heavier one was collected in the sediment. So, these two things was then isolated and was analyzed. So, what they tried to found was, in case of the phosphorus, where it was taken as a inoculating medium, when it was taken in the inoculation medium, it is found in the DNA, and this DNA was isolated in one of the medium where DNA was found in the bacteria, and it was responsible for causing this disorder. So, this radioactively phased protein in the liquid was not radioactive, and in the other case, when the bacteria was infected by the E. coli. So there, this radioactive material was found inside the E. coli body. So that is how here in this experiment also they even tried to prove that DNA is actually the genetic material which is getting transferred. So three things that happened over here was first the infection. So infection is when a virus comes to the host and transfers only the genetic material. Now in case of bacteriophage, it is DNA that behaves like the genetic material. So here this DNA got transferred inside the body of the bacteria. And later when blending was carried out, so this virus was allowed to get removed from the body of the bacteria. And then in the third case, when centrifugation happened, so things got separated on the basis of size. So in the supernatant, this virus body was available, which was made up of protein and it contained that radioactive material, while in case of the sediment that contained the bacterial body was having this radioactive phosphorus in it. Okay, so as we conclude here that the DNA behaves like the genetic material after seeing this three experiment, now when it comes to the properties of genetic material, so what exactly can behave like a genetic material? Why it is not protein or RNA? And what property this DNA needs to fulfill to behave like this genetic material? So as you are aware that DNA has got a capability to generate its own replica. So when a cell divides from 1 to 2, even the DNA gets double. So it has this property of making its own copy, which is only found in DNA. Then it should be chemically and structurally be stable. It should not change just because of any change in the environmental condition. So that is how it has to be structurally and chemically stable enough so that it can get transmitted from the parent body to the new offspring or to the new daughter cell. And also it must provide the mutation with the aging, or you can say with the time, it should be able to get mutated, because that is the only reason by which a new organism can arise, a new change can be found in the existing organism. So that becomes the reason for the evolution. Okay, but it needs to be slow and steady. And the last one and the most important one, it must be able to express itself as Mendelian character. So, as we talked about the dominant and the recessive traits in case of Mendel, it must be able to show the same property. So, these are the properties of genetic material which is completely fulfilled by the DNA and not by RNA or protein, and that is how. So, this DNA behaves like the most appropriate genetic material. We'll take a comparison why it is not RNA. Because when it comes to RNA, it even has the same kind of structure just like DNA, just only it being a single stranded one. But moving to this presence of thymine and uracil. So as thymine is replaced by uracil in case of RNA, that becomes the reason for its instability. And the other point is the absence of hydroxide ion in sugar in DNA makes it become more stable. So, moving to the next topic now, RNA is unstable, but in some of the viruses, even this RNA is found as the genetic material. And some of the examples are bacteriophage, tobacco mosaic virus, and so they are able to mutate fast, and so they are able to evolve faster also. Okay, and talking about DNA, so DNA being the double-stranded one, it behaves comparatively better stable genetic material. In appropriate condition, they come together, even like if they get separated because of heating. But if the appropriate condition is maintained, even this double-stranded can get bound together again. So in this Griffith's experiment, we found some of the property of the DNA that was like a heat killed, in case of heat killed bacteria where the DNA was able to survive, it's because of this property only. It is able to show the stability even in after the condition gets appropriate. So the information in the DNA remains more far better maintained and even stable, and also it is able to get transmitted from the parent's body to the daughter's cell, okay, and it is also able to express its characteristic in far better way. But RNA plays an important role for the synthesis of proteins. So first of all, this DNA needs to be transcribed into RNA, which takes the information in the transmissible form, and then it can later be responsible for the synthesis of protein that actually reveals the property of that particular characteristic that DNA carries. So we will conclude our topic over here. So considering that DNA being the most appropriate genetic material, and so it is found in almost all the organism except for some of the viruses. So thank you.